The title of the presentation is Of Variants, PCR, and Pandemics, the Science Behind. So let me tell you a story, the story of two viruses which are now present in the Philippines, which are creating a lot of problems in the Philippines. One type of virus, which everybody knows, is the SARS coronavirus 2, which affects human health, started coming to the Philippines in around February of 2020. The second virus is the African fever virus, which started coming to the Philippines in 2019. Now, SARS coronavirus affects human health. ASF virus affects human livelihood. And these two are creating havoc right now in the Philippines. One virus comes from wild bats, another virus comes from wild boar or wild pigs. So if you can see, both these terrible viruses are coming from the wild. What does that mean? It means this could be a direct relationship of what humans are doing to nature, wherein viruses who are staying in the wild for years, for centuries, are start coming up. Both viruses spreads worldwide. ASF virus started in the 1900. It came from southern Africa. It slowly goes up from wild pigs to humans to secondary host. It went two ways. It jumped from Africa to South America. Then from Africa goes to the Mediterranean countries, goes to Spain, Germany. Then it goes eastward to the former Soviet republics. And eventually in 2017, 2018, it went to China. Now, this wave, uh, eventually it went down. It went down to Southeast Asian region, went to Vietnam, the Philippines, Indonesia, Myanmar, and uh, and Australia is practically trying to avoid entry of this virus. Now, if you look at ASF virus, it took a century before it reached the Philippines from its from its from its origin. Now, the other virus is SARS-CoV-2 virus. It started in 2019. So it started in China. It started going worldwide as humans carry it. Now, these two viruses also has a similar pathway. If you look at the comparison, the original SARS-CoV-2 virus from wild bats are transferred to humans or even alternative animals. Again, and through that method, it went to more humans. Now, African swine fever from wild pigs were transferred by humans and also by secondary animals like minks, which eventually transferred to farm pig population worldwide. Let's compare now the severity of these viruses. Now, uh, one one virus spreads fast but kills slow. SARS corona cases as of I think as of this month have totaled 128,184,038 with a total death of 2.8 million, and that's around 2.19% mortality. African swine fever virus spreads very, very slow, but it kills fast. The mortality rate is 90 to 100%. Last year alone, 5.4 to 6 million pigs were killed by the virus or culled, meaning they were destroyed, they were killed before they can transfer the virus. That is why I think those in the Philippines right now, you are questioning now why the pigs, the pork product now is very high. This is one of the reasons. In Batangas alone, what we heard of is that many have depopulated their farms. Both viruses produces symptoms within 14 days. Now, there's also a good comparison. SARS-CoV-2 forced lockdown. Lockdowns of cities, lockdowns of schools, lockdowns of factories. And they stresses medical health care. ASF virus forces farm lockdown. Farms are closing. And it stresses veterinary health care. For both instances, it costs money. Costs money, it costs the economy. Now, why are I'm comparing these two types of viruses? Because I I, I have three. Number one is very hard to control. Number two, no really workable vaccine yet. Well, you might be asking, there are many vaccines right now. Right now, yes, that's true. But remember, vaccine to be effective and safe has to undergo at least 3 to 5 years of continuous trial. The vaccine for SARS-CoV-2 have been around, have been developed and been used in less than a year. So you will know that there was, you can read in the news that there's a lot of issues, a lot of side effects, and sometimes it is not effective. Now, these two viruses will stay and circulate the world from now on. Many are hoping that the DNA ASF virus and the RNA SARS-CoV-2 virus will go away. No, they will not go away. They will stay going around in the world right now. The main issue right now is how to slow it down. Okay, why? Why would they stay here? Very simple. It mutates fast. As we speak, there are a lot of ongoing mutations, and those mutations are accumulating. And that is why we are having problems for both viruses. Okay, let me explain further. Maybe what we can do is we have to understand, we have to define now what is mutation. Okay, this one I took from the net. This is a simplified version of how the virus will, will affect or multiply. Okay, if you look at number one, the virus will try to enter the cells. If it manages to do so, it has to be engulfed in the cells, then release its RNA. Now, the RNA right now, you consider it like a computer code or like a program, like Microsoft Word, Microsoft Excel. Now, these programs will practically hijack the cell and telling the cell to reproduce him or reproduce the virus. So basically what it does, it goes there, it's like a bank. You go there, grab the bank, and practically, I want more. So the cells will be overcome, and it has to force to produce all its components. Uh, what is mutation? Now, mutation, I hope I will make it as simple. As the organism or the virus it tries to multiply, it creates copy of itself, whether it's a DNA virus or RNA virus. Now, DNA virus and RNA virus multiply differently. Okay, they use different strategy. Okay, different strategy is like watching those uh, adventure or action movies where there's a bad guy, the good guy try to outmaneuver each other. So basically, the main aim is how to multiply itself. Now, I'll give you this one sequence. A DNA is composed of four different base: adenine, cytosine, guanine, and thymine. Now, it has to act in order for you to multiply yourself. You have to double or multiply the DNA you have or the RNA you have. So it must have or it must come up with the same exact copy. Okay, that's how things work out. Now, a mutation, a portion of that DNA changes. Look, I We are using the same letter, the same sequence. Now, somehow there are changes, and I encircle the change. If you look at the change, it should be CGT. Now it becomes AAT. So it's like you are typing a Microsoft Word and or texting, doing WhatsApp. And when you type your message, inadvertently you will tap on a different letter. Okay, so basically that's how it goes. There are changes in the potential. Now there are at least three different types of changes that happen. Now change meaning modification, meaning it's the same length, the same number, but there are changes in the base. One example, I call it modification from C it becomes A, from G it becomes A. Now, another type of mutation or change is like there is an insertion, meaning an entire sequence, an entire length were added, to the DNA. Another type of mutation is instead of being added, you remove. Now, all this type of mutation has a consequence. It it can be good or it can be bad. Now, let's start with the good one. Good one, it means if I am good one for humans, not for the virus. Good one for humans. Let's say the virus mutates, and it's a good mutation for us. That's for humans. What do you mean good? It means the disease it causes will become less, or stop transmitting. It means it start transferring. The humans are not becoming infected anymore. Or technically, it will stop on its track. Now, you cannot kill the virus. No matter what you do, you cannot kill the virus. The virus will find a way for it to hide. And when the right time comes, right situation comes, it will go back. If there's a good consequence of mutation, there's also a bad consequence of mutation. Now, bad consequence meaning it becomes faster. It multiplies fast. When you say it multiplies fast, meaning instead of let's say 14 days, it becomes 10, it becomes 11. So the transfer rate now increases. It's like suddenly you step on the gas of a car. The virus is driving at 80 miles per hour. Then all of a sudden it goes to 100 miles per hour. Now this is a bad thing because as it transfer or as it jump from one host to another, from one human to another, the rate before the other human will pass it on will become lesser. Will become faster. So less time. Now, another bad consequence is actually severity, increased severity, meaning you become more sick or you can even die. These things are happening for both viruses, the SARS-CoV-2 and the ASF viruses. That's why it's very hard to control. Okay, then it boils out to us now, what are variants? Now, mutations are changes. But you see, mutation is not only single, it's not only one. It's like you are creating a paragraph and you have a lot of mistakes, a lot of typographical mistake in the entire paragraph. That one change makes the virus or the changes in the virus becomes more. This is what we call variants. It starts changing itself. Now, as it goes on, the variance becomes a strain. Now, when you talk about strain, it means there are already physical changes that's happening in the virus. Maybe the spike becomes longer, maybe the spike becomes bigger, maybe the number of spikes becomes more. Then there are already changes in the function of the virus. So some of the functions could be it would fight like HIV. It would fight the immune system, practically destroys the T cell. So there are difference between mutation. Mutation is change. Now, if you have enough mutation, you come up with a variant. The variant right now, although the structure the same, maybe the same, but it has enough change to either increase or decrease the effect of the virus. Now, if we have more changes, the virus itself will change, will modify itself. Now, there are three prevalent SARS-CoV-2 variants right now. I think everybody knows about this: the UK, the South African, and the Brazilian variants. So, what's the difference? Now, it depends now where it mutates. There are you might be familiar with terms like N501Y, E484K, K417N. Those are practically location in the spike protein. When say location, for example, let's say my analogy would be humans. Let's say there's mutation in my arm, my left arm. Another one mutation in my right arm. Another one mutations in my feet. So basically, certain locations in the spike protein of the virus changes. Now, unfortunately, the reason why these three variants are quite being noticed right now because the location of the mutation also coincides in how the virus will bind to the cell. That's the case of UK. So what does this mean? It means that if you have a mutation on this part of the spike protein, the tendency for you to bind more, meaning for you infection rate will increase. So it becomes more infectious. And this is going around 70 countries. Now, the South African variant contains the UK mutation plus two more. And the other two more is that it is not only infectious or more infectious, but it can practically attach to the antibody, reduces the attachment to antibody. Okay, what do we mean by that? Now, if you have a virus or a bacteria, the body, the host, the human body or the animals would have to create a counter. You have to a countermeasure to fight the virus or the bacteria. We call it antibody. Now, it has to attach to the virus so that it can destroy the virus. Now, if it does not attach, then the virus or the bacteria will not be destroyed. And that's the South African variant. So it is not only infective, but it can fight the immune system. Now comes the Brazilian variant. It has all. It has both the UK variant mutation and the South African variant mutation. So far, uh, researchers all over right now try to figure out what are the effect of the Brazilian variant. Now, I think a week ago there was they call it a Philippine variant, which contains all of these. So that's where we are right now with variants. So as you go along, there will be more variants. India last week had announced that they have 441 variants. Now, the moment you hear variants is that it may or may not be infective, meaning it can be a good one or a bad one. So what it means is that for India alone, they are trying to figure out of that 441 variants, how many will be reduced, how many will become more problematic. Out of all the hundreds of different SARS-CoV-2, 27 are currently being monitored all over the world, uh, and they have definitely more than the three mutations or the three types of mutation that the UK, South African, and Brazilian have. All the virologists right now are quite busy try to monitor how these variants will progress. African swine fever is the same. Now, This is a map wherein and there's a circle in each of the area. The circle practically tells you the type of mutations, or the type of variant. Okay? For example, if you go to South to Africa, all the red parts are where the African swine fever are. Now, you notice there are two different circles. So it means that each of the circle there are certain mutations within the virus, which is different from the other area. But there are areas where they are common. So in that common, practically there is a third variant. In Europe alone, there are two two major types. The one in the Mediterranean with the small circles, and the other one is on the big side, where you are talking about Poland, Bulgaria, Romania. Then there's a very big chunk that covers the Soviet Republic, and in Asia there are actually at least three major variants. One covering the Korean Peninsula plus southern Japan. Another big chunk is the Chinese one, and the other one is the Southeast Asia. Now, within this geographical mutation, there are submutations. So there are variants going on. That is why uh, there is a lot of difficulties right now trying to pinpoint uh, how the virus, what are the final, what are the variants, what are the strain of the virus. I just clip a picture of a Pig Progress is a very good magazine for farm, and they said that there is this variation. From our own studies, when we do genetic analysis of the ASF virus from Eastern Europe, from Estonia, Poland, Georgia, there's already a very big difference to the China strain. There is a Normally we are looking at around 600 plus base pair, or those are 600 units uh, for the ASF virus. Now, the China virus, some of the China virus have already a huge deletion, meaning 200 out of 600 will change. So technically, the question right now is that uh, what will it do? Will it become more infective? Will it become more severe? Or the transmission and the infection will go down? Now, the question now is this: variants and PCR tests. If there are variants, will it affect PCR test results? That's a very big question going around right now. Especially if you read the news, I think a week ago, that there is this variant in France that were not detected by PCR. I go to the news, I think CNN and BBC managed to broadcast it, that that is not the first one. There are several cases last year for the uh, that there are changes. Now, technically, then we go to PCR right now. So try to understand whether variants and PCR tests the variants will affect PCR tests. Let's go to the basic of PCR. Now, this one is from the net. So basically, there are at least six different requirement for PCR. Now, if you are using RNA virus, you have another component, which is the reverse transcriptase component. Now, but basically, you have your sample, you have a primer, nucleotides. Okay, nucleotides are basically the building blocks. It's like a Lego set. You want to create a Lego module. You need at least four basic building blocks. Now, Taq polymerase is the enzyme where you can practically uh, produce multiple copies of the DNA or the RNA. Then you have your buffer composed of uh, you know trace base or magnesium chloride and all those things. Now, you need a thermal cycler, a PCR machine. I encircled one. component of the PCR, which we call the primers. Now, what are primers? Okay, primers are like bookmarkers. Now, or if you do Google, everybody's doing Google right now. So if you do Google, right, if you want to search on something, you have to type in, you have to type in a certain phrase, a certain word. Uh, So it means it's your, uh, that's your keyword where you then Google will give you recommendations what they are. No, primers are similar. Primers is basically a marker. No, it is composed of also of a sequence. Now, if the marker, okay, if the sequence, the target sequence, uh, is similar to the marker you have, it means that you have that DNA, and it will bind to that DNA. You can capture that DNA, and you can copy it. So basically, primers are markers. Markers for PCR, you do from the start and from the end. We call it a forward and a reverse prime. So basically, you mark two endpoints, and the moment this primers goes there, these two endpoints will be captured, will be cut, and will be copied. Okay. So uh, this is quite important for our discussion. Now, remember, now primers are to be designed. Now, currently for PCR testing, there are different ways how do you design the primers. It depends on your target. So here, this is a picture of animated cartoon picture of the SARS-CoV-2 virus. It has practically five different sites where you can design your primers. Now, one is the S protein primers. The other two is the M, the E, the R, RNA or RDRP, and the N for primers. So uh, the problem lies is this: if your primers can only detect, let us say, one site, for example, S protein or S primers. If the S primer changes, you may not detect the virus. That's why the key here in the early days of the pandemic, many of the test kit, many of the RT-PCR test kit are only using one or two different sites, and that one creates a lot of issues. There are cases even in the United States wherein uh, I think if you remember February 2020, CDC, the United States Center for Disease Control, had distributed a test kit, and they have to recall the test kit because there was a fundamental problem in the test kit. Of course, they did not say, but it seems that the design of the primers plus the negative controls and the positive controls are not that good. So they have a lot of false positive or false negative. So eventually, I think they were able to remedy the problem. But what I'm saying here is this: it depends on the type of PCR test you have. If the PCR test will only be testing one site, one gene fragment, and that part changes, you will have a false negative, false positive result. So to answer the questions, would variants affect DNA RT-PCR test? The answer is both no and yes. It will not affect because if the mutation of the variants are within one or two sites and your RT-PCR covers multiple sites, even though changes happen in the spike protein, you can still detect the virus because you are detecting other parts of the virus. Now, among all the parts, RDRP, and sometimes the N protein, the nucleocapsid protein, are quite stable, meaning those particular sites within the SARS-CoV-2 does not changes much. I did not say it's not changing. What I say, the rate of change is smaller or is slower. unlike the S protein. The S protein, practically, the change rate is quite high. That's why you have the variant. Now, yes, the variants will affect PCR test result if the mutations of the variants are in multiple sites. It means not only the spike protein, but also the nucleocapsid, also the envelope, and your RT-PCR tests are practically not robust enough or not flexible enough to cover. So uh, these are the variables we're talking about. If the variants will affect PCR test. Now, how about variants and vaccines? Now, this is exciting right now. Basically, there are at least two different types of vaccine. The RNA vaccine, the mRNA vaccine, which is Pfizer, Moderna. The other vaccine, which is the viral vaccine. Let's say viral vaccine, meaning the virus were modified or it is not infectious anymore. So basically, they work almost the same, except okay. Let's say this is a mRNA vaccine. In a way, you are copying an actual virus, but the instruction is very limited. Okay, the whole genome of the of the CoV-2 SARS-CoV-2 is 30,000 base pair. No, 60,000 base pairs. No, you only cut or you only get a small portion of that. Then you have to envelope it with a fat, a lipid membrane. Because this mRNA, if it is not protected, it will be destroyed. At room temperature, it will be destroyed. So you need a lipid layer or a fat layer. Now, you inject it, and practically it will be taken inside the cells. Now, the mRNA will be released and practically give instruction to the cell mechanism. Now, there are many questions right now about this approach. So because I think in the news there's a lot of side effects now. All vaccine have side effects. It depends right now how severe the side effects are. Now, consider this: in order for a vaccine, in order for a vaccine to be totally safe or even 90% safe, you have to remove all the excesses in the formulation. That's why it takes years. It takes 2 years, 3 years to do that. Now, this one is less than a year. So it means that many of the other things are still being studied, still being developed. Now, so side effects could be the result of the RNA itself, because maybe it's producing more than it's intended, or the lipid layers. How about the normal viral vaccine? Normal viral vaccine, basically you have a virus, you kill the virus. In a way, it is not effective anymore. All you want is actually the covering or the spike. It goes inside the cells, practically tells the cell to multiply. No, okay. But there is a common denominator between mRNA vaccine and viral vaccine. What are these denominator? Number one, when you create a vaccine, you are basing by a vaccine, you are basing it on either a certain strain of the virus itself. Now, look at the timeline. If it is being rolled out the last few months, so it means the basis for the viral vaccine or the mRNA vaccine was the viruses or the variants in the early days of the pandemic. Now, the virus is mutating. Variants after variants after variants are coming out. What does it mean? If the target of the vaccine is the S protein, and the S protein are the one changing, which creates the vaccine, existing vaccines right now will have reduced potency. And this one was already announced by all the manufacturers. One example, I think Moderna said Moderna said that they have a reduced potency against the South African variant. AstraZeneca is the same. I think Pfizer had created a modification because initially before the UK, they cannot their potency had reduced if there is a UK variant. Here's the problem: there are variants coming in and out and in and out. So the main issue right now between vaccine and variants is that it's a how do you call this one? It's a continuous chase. It's a continuous chase. There are more variants. The vaccine will not work. We have to change to cover the other variants. Then new variants will come in. Then you have to change the vaccine again. So this is what's happening with influenza. Remember, if you have influenza jab, every now and then the influenza vaccine you have to be vaccinated again for another type of influenza. The only problem with influenza, it does not mutate as fast as coronavirus. So, we all know that vaccine will not give you full protection. No vaccine currently available will give you full protection for the simple reason that there are variants coming every now and then. Now, the question now is, what will the vaccination, what's the purpose of vaccination? The purpose of vaccination, it now becomes a risk management uh, scenario. Is either you want a protection, even how low it is, or you don't have a, or you don't want a protection at all. I call it the choice is yours now. So it is not an issue right now where I will be protected or not. It's how much am I protected. It's either 10% is better than zero. 20% is better than zero. Now, how to end the pandemic? I think I'm in consonance with others when you say three things only. Number one, the first and foremost is crowd control. We have to limit the transmission of the virus. Because remember, as the virus is being transmitted, as it's being transferred from one body to another, it always mutates. If you want the mutation to slow down, you have to limit the transmission. This is not an easy solution, but it can be done. Now it depends on country per country. Here in Singapore, they said that because Singapore is a very small place, that the government was able to do that. So basically, local transmission here are very reduced. The only issues right now is when Singapore starts opening its borders, because there would be new people coming in, and new variants. We expect new variants. Now, another one is early detection. When I say early detection, I'm not only talking about PCR tests. What I'm talking about is a mechanism. How do you do early detection? Uh, it is a system. It is not. It is not whether you do antigen test, antibody test, or PCR test. No, those are tools. You have to make the tools into a system so that you can create a robust protocol. And third is actually achieve immunity. Now, there's a big question mark on this achieve immunity because, as you all know, as there are more variants coming in, the vaccines that you have or will be given to you may not be effective. So you cannot end the pandemic with just doing one of the three. You have to do all three, and that's where management comes in. So I hope this presentation provides some clarification. Remember, I think it's the obligation of UP, faculty, staff, students, and alumni alike to provide the truth and correct the fallacy. And thank you very much.